Hey guys, thanks for coming back. Just kidding, I know you have to be here now. So today we're going to be looking at the second video in our genetic mapping series, the 2-point test cross. So what is a 2-point test cross? If we think about this with respect to the physical maps, this is Hyrule again from the first video, think about 2 points being exactly that, 2 different points on the map. Okay, so last time we used Hyrule Castle and Recombination Prairie. Okay, so these are 2 points. And what we're going to end up wanting to do is figuring out how far apart these 2 things are. Okay, and that seems pretty straightforward. You can count this. Okay, they're about that far apart. But the thing is, when there are only 2 points, what we don't really know is where everything else is in relation to them. We don't know, are they really this close together? Is this guy really over here hanging out? So when we only have 2 points, it's really difficult to actually say how far apart they are. Okay, so when we think about these 2 points, how are we going to measure this distance? Okay, and so again, we can't really, when we're looking on chromosomes, just kind of eyeball it, look under the microscope. Oh, this looks like it's about this far apart. There's no real indication where these genes are necessarily going to be. So we have to think kind of indirectly. How would we measure this distance between our Hyrule Castle and Recombination Prairie? Can we measure this with GPS? No, we don't have any information. Are there roads here? I don't know. What's the terrain? Are we going to take a flight? How are we going to get there? And what units are we going to measure this in? Are we going to measure it in the time it takes for 0.3 blood moons to rise once again? Are we counting this how long it takes to get there on foot? What if we take Epona? Will it be a little bit shorter? And again, using paraglider, checking out that stamina wheel, we could think about doing this measurement in a lot of different indirect ways. Okay, when we think again about the chromosomes, how does this relate to the chromosomes? We're going to have to also think about kind of an indirect measure. So again, in present day, we know that if we really wanted to, we could probably just take a look at the genome sequence and figure out, okay, this is exactly how far. That would be equivalent to, again, looking at a map and looking at the actual mileage that it was going to take to get there. But again, when we started genetic mapping, sort of the first experiments that were done, and even now it's useful, we have to think about this sort of indirect measurement, okay? So for chromosomes, our indirect measurement is going to be recombination rate. So we talked about recombination in the last video, so just to refresh your memory, we're going to spend a lot of time on the details of this recombination rate today. Again, we're looking at this crossing over where during meiosis 1, we can actually mix and match this genetic material, okay? So again, let's get into the actual chromosomes. Let's see what's happening here. So we're going to ultimately be looking at 4 different recombination scenarios that can happen during meiosis. What we're going to be focusing on is what kind of gametes we're actually making. Okay, so here we have the parental genotypes. We know last time we looked really intently at parental phenotypes or recombinant phenotypes. Okay, so we had these 2 things. And we're really thinking about, are these going to be in the same sort of coupling with the parent, or are they going to be out of sync? So those are kind of the details that we're looking at. Okay, so here, again, we're focusing on the chromosome. Last time it was 100% about what's going on with the phenotype. Now we're really getting in there to the chromosomes. Okay, and you can see that we're going to end up in this scenario having 1 parent that's going to be wild type at both of whatever these genes are, whatever traits they're for, wild type with wild type. And our second parent is going to be recessive with recessive. Okay, so we have this set up for our parents. Okay, of course, as parents do, we're going to make some gametes. Okay, and so we're going to first concern ourselves with the most basic idea of 1 single crossover event. So this is really not going to be different than anything that we've seen, but we're going to use this to work our way into things that are a little bit more complex. Okay, so again, what we're worried about is going to be the status of the products, aka the gametes. Okay, so thinking about this, we can actually sort of take away this sister chromatid and this one down here, because we can already see that they didn't even actually participate in this crossing over in any way. So we know that they're going to stay coupled. We're going to have wild type with wild type here in our product, and we're going to end up with recessive, recessive here in our blue product. Okay, and so just to take the mystery away, our resulting genotypes, obviously we are coupled and coupling, we look just like the parent. Okay, so what's going on down here? Again, this is a very straightforward case. We have some solid recombination happening somewhere between these two genes, and what we end up with is going to be this decoupling. We're now going to have our wild type A and our recessive B on our first gamete, and then we will have recessive A and wild type B on our second gamete. Okay, so thinking about that, how are these guys going to plinko out? Okay, so thinking these guys are going to be recombinant. They're going to show, if we're thinking about phenotype, the recombinant phenotype, and looking at the chromosome very clearly, recombination has occurred. Okay, so we can think about the idea of these 2 points. This is the 2-point of the 2-point Testcross. Again, thinking about this like those 2 points on the physical map. Okay, so going back to our map in Hyrule, again, remembering that when we started, all we had were these 2 points, and it was a little difficult to tell what's going on in this distance. Okay, so even though we are #blessed and can see the entirety of this gamete, okay, we can see where yellow goes to blue, blue goes to yellow down here. Remember that these 2 points, as we're working out this experiment, we actually will only see the places that we think to check. We won't actually see what's going on. Remember, we're not looking in a microscope. Instead, what these results will look like, going to be a PCR assay, something we run on a gel, CODIS markers, again, like the FBI uses to identify people. These are going to be the places we check. And so that is going to come into play a lot later on. We're going to see it more in today's video, and then we'll see even more of this when we start to expand this out to the whole genome. Okay, so remembering that idea that when we talk about these 2 genes, whether recombination has occurred here or not is kind of a moot point. When we talk about this being recombinant, what we need to see is definitively the decoupling of the 2 points that we're specifically interested in. Okay, so that may seem kind of weird right now to spend time on, but again, as this gets more complex, I think it'll be important to note. Okay, so let's take our second scenario, and you can already see some stuff's going on. Okay, so we're this time going to see a double crossover occur. Okay, so we're going to have one recombination event, and then somewhere else between these 2 genes, we'll have a second recombination event. And you can see we just sort of exchange this little chunk right here. Okay, so that's neat. We can kind of observe what's going on. We're again going to pay attention to the gametes, because really that's what's going to be important. Okay, so checking it out, we can see that this gamete is actually going to still just be coupled. These 2 traits are coupled just like in the parent. This guy down here, these are not participating in this double crossover, so we can go ahead and kind of not think about them, go ahead and sort of list these as being parental relative to these 2 genes that we're looking at. Okay, so thinking about that, we have to remember that what we're going to look at is what's going on here. So we do have crossover events occur. You can clearly see as we're looking along this chromosome, crossing over events have occurred. So what is that going to mean when we categorize these 2 gametes? What is that going to mean? Okay, so this, I think, is a really good time to pause that video. Phenotypically, are the 2 remaining gametes that we're talking about, are they going to be categorized as being recombinant or parental? And what do you guys think about the genotype as well? Okay, so I know that you really spent a lot of time thinking about how to categorize these. This will be really important when we start to get to the 3-point crosses. So let's again kind of review what's going on here. So the important point, which we looked at in the single crossover scenario, is really the important part is where did you check? Thinking about, we care about these 2 points. We don't really care, except in terms of distance, what's going to be happening in here. I don't care if I had a bunch of crossovers. I care about the status of these. And again, we may have only checked there. So if we just check those places, then again, that will be all that we see. Okay, so we can go ahead and kind of like focus in on that. And again, you see that we have our wild type A, wild type B still in coupling. Okay, so here we have our recessive A, recessive B still in coupling. So regardless of any crossovers that actually happened here in the center, the recombination that happened, we cannot account for it because we only can see these places. So that makes these gametes the parental gametes as well. So you can see in this scenario with a double crossover, all we did was exchange some material in the middle and end up being parental. Okay, so it's about to get wild in chromosome world. When we look at our next scenario, we're going to see a case with a 3-strand double crossover. Okay, so yeah, this is a 3-way between chromosomes. Don't come at me. I didn't make this up. Okay, so what we're looking at on the top, this is one way that this could happen. So you can see kind of who's involved here. And then a second way. So we're kind of lumping them together in terms of, okay, it's 3 strands being involved here, but only one of these is going to happen at a time. So remember, biologically, we're only producing 4 gametes. Okay, so either you did this top scenario and got these 4 gametes that we're going to look at, or the bottom scenario. So we're not getting 8 gametes. While I see why that might be an evolutionary advantage, currently that'd be some kind of full DNA alchemy. So we're not going to visit that. Okay, but we can look at both potential cases. Okay, again, just for simplicity, kind of taking out of the picture anybody that doesn't participate in this. So you can see we've got 3 players in either scenario. Okay, so again, that's synaptonemal complex that forms. It looks like there's space to spare. There is not. They are very tightly connected, so this kind of thing is possible. Okay, so we can think about how this is going to play out. All right, we know those top ones are parental, so there we go. So now we just have to think about the rest of this. Okay, and so you probably know about what's going to happen. We're going to focus on the points that we care about, the 2 points for this 2-point test cross. Going to be coming up. Okay, so again, kind of thinking, let's not even worry about what happened in the middle, who is with whichever chromosome. It's not really going to matter. Again, what we care about is what's going on. Okay, so you can see here we ended up wild type A with recessive B. That's going to be recombinant. We're going to have recessive A and B like the parent, and then another recombinant here. In the second scenario, recombinant, decoupled, wild type with wild type, parental, and then another recombinant here. Okay, so in our first scenario, we end up with about half and half going down here. Okay, so we end up with about half and half. Okay, and so in our next case, our last one that we're going to look at, and these are all the potential things that could happen. Okay, now we have a 4-strand double crossover. Okay, so already kind of blank these out for us to think about. Okay, and you can see that pretty much here everybody's decoupled. Okay, so we can end up again thinking, okay, where did the crossovers actually happen? We had 4 players here. We can see that there was recombination between them, but what we could actually see, we end up with decoupling. So we end up recombinant, 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 recombinant. Okay, so all 4 of these. And again, you're making 4 gametes at a time. All right, so one of these 4 scenarios could be happening for any of your chromosomes at any point. but we're just worried, what are the outcomes? And so as we think about what are the outcomes, we've had different proportions of parental gametes come out and different proportions of recombinant come out for each of these scenarios. So thinking about that and kind of relating this back to recombination rate, let's take a second and think about, can we actually get a recombination frequency that's greater than 50%? So yeah, let's pause that video and think about, are we ever going to be able to get a recombination frequency that's greater than 50%? Welcome back. Thanks for all your hard work on that. So what we're going to be thinking about, again, is can we end up with greater than 50% recombination rate? Okay, so let's add up all the scenarios that we had, starting with our single crossover. Okay, we had our two-strand double crossover, we had a three-strand double crossover, and a four-strand double crossover. So again, what we care about is the gametes, care about whether they're recombinant or parental. So let's just take a little tally up here. How many are parental and how many are recombinant for each of these scenarios? Okay, so you can see 2 of each for our single crossover. You can see that in our double crossover, the two-strand double crossover scenario, we ended up all parental, no recombinant. Okay, for our three-strand double crossover, remember there are 2 ways to do this, but we only get 4 gametes at a time. I think we get parental recombinant, so that's 2 out of 4. Same scenario here. So we kind of lump these together, 2 parental, 2 recombinant, or kind of a half and half sort of a situation. Okay, so then we can think about this last guy, this 4-strand double crossover, and we see that all of these are recombinant. That gives us 0 parental and 4 recombinant offspring. Okay, so again, thinking about the recombination frequency, this is just a percentage. There's no reason to be really afraid of this math. This is really just a percentage. So we're going to think about how many total do we have here. Okay, so we've got 8 on one side, 8 on the other, for a total of 16 different gametes that we've explored here. Okay, and we can see that the recombinant ones, there's going to be 8 of those guys. So that's going to give us 8 over 16. That percentage that we're going to be looking at is 50%. Okay, so no matter what we do in this recombination sphere here, 50%, that's what we end up with. We're never going to get extra recombinants on the whole. It has to be 50%. So what does that actually mean? What is 50%? Why is that the magic number? I think looking at this on a map is probably going to be more clear. So what is 50% on a map? Again, thinking about this physical map that we're working with. Okay, let's bring back Hyrule. Okay, so if you have a 50% recombination rate on a chromosome, what you're really talking about, if these 2 traits are on the same chromosome, is that they are very far apart on that chromosome. So something way down here, eventide, not going to go there, okay, and something way up here. So these can be very far apart if we're talking about on a chromosome physical map. 50% could mean that. Additionally, we could be looking at a scenario where those 2 traits are actually on different chromosomes. So if you remember from the first video, we're talking about linkage, talking about mapping. We have to kind of stick to one chromosome at a time. If we're seeing close to a 50%, near 50% recombination rate, we could be talking about a different chromosome, which is kind of suspiciously sounding like independent assortment. So that would really be what we were looking at if we were sitting around 50%. So really either as far apart as they could go, there may be a couple of other things that could influence this that we're not ready to talk about yet, or sitting on a different chromosome. Okay, so now it's time, since we've covered sort of the basics, to think about how to actually perform the 2-point Testcross mathematically. Let's kind of apply what we're thinking about here to a real chromosome. All right, so how do we actually do this? How do we measure this? So this is a figure from your textbook. Okay, so this is from the textbook. They are outlining this whole cross. This is the whole setup. Again, we're talking about setting up a test cross. Okay, and here instead of phenotypes, they're showing us the chromosomes. But I do think it's helpful, since we're talking about phenotypes, to at least kind of relate this to a phenotypic situation. So let's go ahead and get the chromosomal stuff out of the way, and then we'll go ahead and do that. Okay, so you can see here, this is just the original parents, wild type wild type for both of these traits. Other parent, we're looking at recessive for both traits. Okay, so that's what we're going to see. We're going to cross these two individuals. Okay, and then we're going to end up with our F1 generation. So the test cross that we're actually referring to is not this. This is not the test cross. This is the test cross we're talking about. The double heterozygote, meaning heterozygous for both traits, one wild type copy, one recessive copy, okay, for both of these traits. So this is going to be a double heterozygote crossed with a double homozygous recessive individual, okay? So that's what we're going to be looking at here. And so again, to relate this back to the phenotypic scenario, let's bring back those flies from the previous video, because let's be honest, geneticists be like flies before guys, okay? So flies all the time. We're looking again at this case, looking at maybe some parents that are going to have the gray vestigial wings, or it doesn't matter. This is just to relate it back. But when we really get back to this test cross, what we're thinking about is an individual that is going to look like the wild type for both traits. So this one's going to have the gray body, which is wild type, and wild type wings. And then our test cross parent, we're going to be looking at, again, having that black body and vestigial wings. Okay, and so the second kind of part of this is thinking about the offspring that this should yield. We learned last time that setting up those expectations for what actually is going to happen, that's the important part. We got to get those expected ratios. So we know that this cross should yield 2 parental phenotypes that are approximately equal and 2 recombinants that are approximately equal. Okay, so if we think about the way that that did plinko down in our previous table, it's that same one we used to do the chi-square last time, okay, you can see that again, our expected number, our parentals, and our recombinants, we should end up with a 1-to-1 ratio, okay, and again, that is expected. Observed is what you observe, and you cannot do anything about that. There's no math involved there except counting, okay. So again, we learned about recombination frequency through the previous slides. So we're going to now start to use recombination frequency to calculate relative distance, okay? So the thing that I think is important to note here is that we are kind of using phenotypic information in this case as a stand-in for what's directly happening at the chromosome. So at the chromosome, we want there to have been a crossing over event, but we may not be able to physically check that itself directly. So here we're kind of using the phenotypes as a proxy for directly looking at the chromosomes. And that's why we actually care about the number of offspring that come out with these decoupled phenotypes, where the traits are decoupled from the parents. Because we can easily look at flies and see what color they are, what their wings look like, pretty quickly. That's a pretty quick check. So we're using that information as kind of a stand-in for checking the chromosomes directly. Okay, so that's what we're going for there. We saw in the previous cases that there can be some times where there can be double crossovers and things that can kind of mess you up, but we will get to that when we talk about the 3-point test cross coming up next time. Okay, so again, we're going to get ready to calculate this. Not going to be bad math at all. We're just going to be looking at the percentage of recombinants in this population. Okay, so that's going to be our recombination frequency. So when we think about how to actually math this down, okay, we're going to be 524. We're going to divide that by the total number. Okay, and yeah, I made a little typo here, but it's all right. Okay, we'll probably live through it. So 525 or 524 if you actually don't make a typo in your calculator. Okay, over the total, that's just a percentage. And we can just go ahead and directly convert that to either map units or what is going to be called centimorgans. Okay, so I like to use centimorgans. I don't know if it's old school or not, but they're interchangeable. Okay, so what does that mean for the chromosome? So we've calculated the percentage. You can see pretty directly here. 524 over the total number. Again, using the phenotypes as a stand-in for having directly checked what's happening at the chromosome, we end up with a recombination frequency of 16.2, looking at 16.2 centimorgans. So yeah, let's kind of put that onto a chromosome. So again, let's kind of keep dealing with these fly traits. Pretending body color is up here, whatever gene that is. And our gene for wing size is somewhere here. What that 16.2 means is that the distance, the relative distance in centimorgans or map units between these two genes is 16.2 centimorgans. Okay, so really to just kind of wrap this up, what we've done, we're really preparing for the 3-point test crosses and actually constructing maps out of entire chromosomes, expanding this to the genome. Once we've done that, that's when we can start to actually find those more like destination traits, like those Hyrule Castle types of traits. Okay, so in conclusion, and see that genetic mapping, we're going to be using recombination frequencies to determine relative distances. Okay, so we're going to see there might be a little bit of a difference between what we get for a relative distance in map units versus what's actually physically going on the chromosome. Okay, the physical sequence of the chromosome, the physical length, that again is like looking at a map. Okay, physically opening up your GPS, opening up Google Maps, and seeing, okay, this is how long this is how long it will take to get there, telling you the speed traps. This is the whole thing laid out. We're still dealing in relative distances. Okay, so the other thing we can really get out of this is that our crossing over events can be estimated. So these actual recombination sort of tradesies of DNA, we're estimating these by looking at the phenotypes of the offspring. So that's why we've been spending so much time thinking about the phenotypes of the offspring. And then we learn to calculate this actual distance, taking a percentage, recombinant offspring over the total. So next time, we'll be looking at 3-point test crosses and really expanding this out to the genome. So go ahead and end this with our joke of the week, and don't forget to like and hit subscribe. Just kidding, I know you have to be here.